Good afternoon, everyone. Thank you and welcome back. Thanks again for joining us on our virtual Space Fest adventure this year. As many of you know, this is one of our favorite events come every winter. And although we aren't able to do it with you in person, we have a ton of activities online and lots of talks with scientists and researchers over the next few days. We would very much like to thank Ruth and John Huss and their generous support of Minnesota's astronomy classroom as we continue with these programs. I'd also like to encourage you to check out our website. There are lots of Space Fest things to do and even some online activities where you can complete projects and submit what you created to us online through social media. Right now, we're moving on to our next adventure, and I'm happy to say that I'm joined by two of the Bell's own. Adam Hartman is our gallery programs assistant, and Sarah Komperud is the planetarium programs coordinator. And they're here to share a special critter with us underneath the microscope and talk a little bit more about its special astronomy connection. What's our adventure? What are we going to look at today, Adam and Sarah?
Thanks, Amber. So we're going to do another microscope challenge type thing today. So for those of you who have joined before and done our microscope challenges, I've usually used specimens, which are not alive, and they're much bigger than what we have today. So I typically give you a clue and I'll zoom in and out as we get further along so you guys can make guesses. This particular microscope challenge has a live organism that is very small, so I'll keep it on a constant magnification and give you guesses the same way I normally would. So I'll switch over my camera so you guys can see what we're looking at. And hopefully everything looks good. You should see some squirming. Amber, Sarah, do you guys see that? Okay, excellent. So what I'm going to do is I'll give you a series of 3 clues, and in between each clue, I'll have you guess what you think this organism is. So your first clue is, this organism has 8 non-jointed legs, each with 4 to 8 claws that help them cling to plant matter. And hopefully they stay in focus as you're making your guesses. Do you have any guesses, Amber?
We have some very smart audience members. I have someone that says, bet it's a tardigrade.
Well, I won't say anything yet, but I'll give you your second clue. Your second clue is, many species feed primarily on juices from moss, algae, and lichen, but they sometimes eat each other and other microorganisms such as rotifers. And for those of you watching, this is at about 200 times magnification, and this is as close as I can get with the microscope I'm using. So hopefully everything's still in focus. I'll give you a third clue. Their Latin name translates to slow-paced or slow stepper, but they are more commonly known as water bears or moss piglets. And with that, I'm sure some more of you have probably made the correct guesses.
I do indeed see that, Adam. It looks like we've got a couple of guesses. We've got caterpillar, we've got another guess of tardigrade, and then our all-time favorite of moss piglet.
Awesome. So it was or is, there are some tardigrades in there, moss piglets or water bears. They are swimming around eating some algae and just hanging out. Are there any questions?
I haven't seen any questions come up yet, but I will jump back in and let you know if the audience has any questions. Sarah, do you have some fun stuff to tell us about water bears or tardigrades and their connection to astrobiology and astronomy?
Yeah, so the fun thing that I didn't even know about before looking into this is that they don't necessarily live in just the extreme environments that we've heard of, like the really hot and the really cold, the vacuum of space, or deep sea pressures like the bottom of the ocean. They actually quite just like being in a nice thin layer of water and eating their moss. So Adam, I believe you have even found them living out in our pond here on our landscape at the museum. And so they are quite common from what I am gathering here. And in fact, what I also didn't know is that there are over 1,000 different species of tardigrades. Now, what we're talking about with them and what makes them so extreme is their ability to survive in those hot and cold temperatures, the high pressures, the high doses of radiation out in space and the vacuum of space. They've been actually found to survive at temperatures of minus 328 degrees Fahrenheit, all the way up to positive 300 degrees Fahrenheit. So that is way larger of a range than any of us would be willing to withstand. And at the pressures of the deep sea ocean that we found them at, those pressures actually would turn the proteins, the DNA of other animals, and just kind of start ripping up that DNA. And so that makes us wonder, what is it about these creatures? How can they survive in such extreme, such different types of environments? Well, it turns out what they can do, especially in really cold or dry environments, it's called cryptobiosis, where they can squeeze all the water out of their bodies as their environment is drying up. Because if they're living in a pond or a puddle that might evaporate really quickly, they need a way to protect themselves because they do need a thin layer of water to survive. And so this process of cryptobiosis will actually help them squeeze out all the water of their bodies. They'll retract their head and their limbs and curl up into this ball here called a ton. So this dehydrated ball, and they kind of become dormant here. And studies have shown or have tried, and they've been able to come out of this ton after 8 years. Now, some studies claim they were able to get longer, like 10 or even to 30 years of successfully coming out of this kind of desiccated state here. But what's been able to be repeated is around that 8-year mark right now. But when they dry out, they actually create this kind of sugary glass substance that inside their cells, so that way their cells don't become destabilized, or any water that's left in there, if it starts to slowly warm up, the water doesn't quickly evaporate. So they have this way to protect their cells as they kind of go into that dehydrated state with this gel-like sugary substance, which is really interesting. And another thing that we're thinking about is how can they have all these different environments they can live in and all these different types of ways to survive them? Well, it turns out that they might have something in common of all these different environments they have been known to survive in. So the extreme heat and cold, radiation, high pressures, those are all ways that DNA gets damaged. And so what we're trying to figure out here, or what scientists are trying to figure out, is how are they repairing their DNA? And so maybe it's one common defense or one or two common defenses for them that make them able to survive in all these different types of environments. But what we've also found is, and I found really kind of surprising, with all those different species of tardigrades, the oldest and most primitive one, they call it Anthrotardigrada, can actually not survive, and they can't do all these different environments. They are coming from the ocean, which suggests that once tardigrades left the ocean, came on land to live in smaller pockets of water, that transition is where they started to evolve these defense mechanisms that allowed them to survive in such extreme environments. Now, what are we doing with all this data? And what does this have to do with space? Well, I'm sure you've all heard that we've launched tardigrades into space and they've survived. A small collection of them went up in the Tesla a few years ago when Elon Musk launched this Tesla up into space. They were in the glove compartment. But they've done some studies on the ISS as well, where they sent up a batch and they exposed them to outer space. So the vacuum of outer space, the radiation, some of them they protected from higher doses of radiation, some of them they didn't. And when they brought them back to Earth, they actually showed that about 68% of them had survived, and some of them had even laid eggs in space, and those eggs became viable and healthy back here on Earth. And what we're trying to do this is figure out, how does this radiation interact or damage our cells? Because if we're going to go into space for long periods of time beyond low Earth orbit, radiation is actually one of the primary, if not the primary concern for keeping our own human bodies healthy and safe when we're trying to, let's say, fly to Mars and back. There's a lot of extra radiation outside the protection of Earth and low Earth orbit that we'll have to deal with.
So we did have a few audience questions that come in, and some of them are on a little more on the biological side. Tanya is wondering a little bit more just about how you describe how small they are, and April is wondering a little bit more about how they fit into the ecosystem. Do either of you have any thoughts on that?
I'll leave that one to Adam.
I guess it's hard to say how small they are. They're microorganisms, so at least with the jar that I have, I am unable to see them with the naked eye. I see the clumps of algae that they're eating, but I can't see them without looking at them under a microscope. I think that there are some larger species that can get up to like a millimeter, maybe a little less, which is still pretty small, and it's not something you would just see if you were to look into a pond. But they are very small. And what was the second part of that question, Amber?
They were just wondering how they might fit into the ecosystem, like especially the example of us having found them in our own ponds at the Bell Museum.
Yeah. So I think they're just kind of, they obviously play a role, but it's hard to tell what kind of role they play because they're so small. They're not like a really great food source for any large organisms that we have in a pond. But you know, everything is food for something, so including each other. So some of the tardigrades eat smaller species and stuff like that. So they have a role in the food chain, and they're exceptionally hardy. So they have value for our own studies.
Jennifer is also wondering if there's anywhere that they cannot survive.
They're pretty hardy. What I found out is that one of the ways that they might die is being crushed. They do have a little bit of a, and Adam, you're probably better at this from the biology side, kind of, I don't think it's called an exoskeleton specifically with this type of animal, but kind of an outer shell, some sort of a little bit more like a grasshopper is I think what I read. But they are still very, very susceptible to being squished.
Yeah, so they do, they go through ecdysis, which is the process of kind of shedding their exoskeleton. It's a little different than like an insect, I think, but they do have different growth stages. And they're relatively closely related to insects and arthropods, but they're not in that same group. So it is kind of interesting that while they are so hardy, they can be physically crushed because they, like an insect almost, with their exoskeleton.
All right, and I have one final question, and Grace is asking, are they from space?
I have no indication that they would have originated from space, but we do know they can survive in space based on our studies on the ISS. There was actually a collection of them that were on the Space IL Beresheet lander that the last moment crash landed on the moon back in 2019. Now, there's no risk of really contaminating the moon because of the way they were sealed into that spacecraft. And then in order to come out of their dormant state, they would need some water to kind of essentially rehydrate. So we know they can survive in space, but they do need water. So I don't know if there's any indication they came from space, but maybe one day we'll bring them back and have them survive.
All right. Well, thank you so much for sharing those those images and the microscopic view. They're very tiny, so obviously, you know, we saw them just kind of moving around a little bit, but sharing all that information about tardigrades or water bears, both from a biological perspective and an astronomy perspective. I hope everyone at home is giving a little bit of thank you to both Sarah and Adam for joining us today. We have lots more fun Space Fest stuff coming up. I hope that we'll see you once again at about 2:30 for our next talk. We're actually going to be talking to a space scientist who works for Goddard Space Flight Center, and he was part of the Cassini missions that studied the Saturn systems. And so he's going to be taking us on an adventure to learn a little bit more about what they were looking for in the moons of Saturn. So we hope to see you all then. Have a great afternoon.